All right, you can see my screen. Okay. Okay, so before I get started, I want you guys I'm gonna explain the TCA cycle and the electron transport chain. And I did see bioenergetics was also explained. So I'll go over it, yeah, like briefly over it. So as you can tell, my name is Khaled. I'm a second year medical student. And yeah, let's get into it. So if you do have any questions, you can either type in chat or just unmute yourself with me. You can speak. It's fine. All right. Okay. So before I get into it, I want you to understand three main concepts. Okay. They're gonna help you understand what enzymes are, what the actual meaning is, and the whole thing about Krebs cycle. So first thing I want you to know is when you hear the word dehydrogenase, okay? Dehydrogenase, as soon as you hear the word dehydrogenase, the first thing that pops in your mind is it forms NADH or FADH2. And I'll explain why that, okay? I just want you to know that for now. Another thing is decarboxylation. Decarboxylation, it's another thing. It's decarboxylate, okay? Decarboxylate. So it releases CO2. So from the actual name, you know what it means. Releases CO2 and dehydrogenase forms NADH and FADH2. Okay. Now, like the previous PAL session, the glycolysis was discussed. And I want you to correlate the previous session with this session in which I'll be explaining TCA and the electron transport chain. I want everything to be correlated because this is how you understand the entire thing. Okay. So like the main point of the whole thing, obviously, it's to produce ATP, right? Keep this at the back of your mind because if you do understand that you can solve a lot of things. I know it's self-explanatory, you do know that, but you have to know how to apply it, okay? So obviously through glycolysis, you produce two ATP, two NADH, and two pyruvate. Now, this pyruvate, because we want to use this pyruvate, right? So this pyruvate, obviously, as we know, glycolysis happens in the cytoplasm, okay? So this pyruvate migrates to the mitochondria, to the matrix specifically. And what happens is there's going to become a reaction, which is this reaction over here. And this reaction, it's not really a part of glycolysis or the TCA cycle. It's just a link in between them. Okay. So this reaction is pyruvate becomes acetyl-CoA. And as we know, for any reaction that occurs, there has to be an enzyme that controls this reaction. And this enzyme is pyruvate dehydrogenase. So how do you remember that? So we know that the substrate is pyruvate. So pyruvate. And it's a dehydrogenase. And if you do remember what I said in the beginning of just at the beginning, I said dehydrogenase forms NADH or FADH2. So at this case, it forms NADH. Now it says two. Why does it say two? Does anyone know why it's two and not? Okay, I'm discussing every time anyone talks about Krebs cycle, it's always like one pyruvate goes into becomes one acetyl-CoA, then enters the Krebs cycle. So why is it two right here? Does anyone know?
Is it because two pyruvate enter from glycolysis?
Yes. Okay. So because we know glucose is 6 carbons and pyruvate is 3 carbons, so it forms two of these. So 3 plus 3 is 6. So it forms two of these. And each one, so for example, when I said here, 3 NADH, 1 FADH2, and 1 ATP, it's per 1 pyruvate. So at the end, because obviously we produce 2 pyruvate, you're going to multiply the entire thing by 2. Okay. We're going to get into that. I just want you to know these for now. Okay. So as I said, pyruvate dehydrogenase. Pyruvate is a substrate, so pyruvate and dehydrogenase, as in it produces NADH. Now, if you do realize, it also produces carbon dioxide here. So it releases a carbon. A carbon is released from the body. Okay. So what that means is that this reaction is also so it's not only a dehydrogenation, it's also a decarboxylation because it releases CO2. Okay. So from the enzyme, we know what it does. So from the enzyme, we know it releases NADH, and we also know that it releases CO2 because it's a decarboxylation reaction. Okay, so 3 carbons becomes 2 carbons because of the CO2 which is released, and produces NADH because it's a dehydrogenase, and as a result, it forms acetyl-CoA. Okay, now we know, as I said at the beginning, we said that the main point of this is to produce ATP. Okay, so let's say I have a lot of ATP in the body. Do you want this reaction to occur anymore?
No. No.
Okay. Okay, so that is why a lot of ATP can inhibit this reaction. And when you inhibit this reaction, the Krebs cycle will not occur anymore, and no more ATP is going to occur. Okay. And another point is, why does acetyl-CoA and NADH, why do they inhibit pyruvate dehydrogenase as well?
Because they're the product of pyruvate dehydrogenase.
All right. So because they're the product, like negative feedback occurs, and they inhibit pyruvate dehydrogenase. Because let's say you have a lot of acetyl-CoA or a lot of NADH, you don't want to form, you don't want to make more, so the products are going to inhibit that. Okay. So I hope this.
I have a question. So pyruvate dehydrogenase also needs lipoic acid and thymine and
Yeah, I'll get
FAD.
into it right now. Yeah, I'll get into it right now. Okay, so as discussed, all right, so pyruvate dehydrogenase, obviously pyruvate enters the mitochondria, it becomes acetyl-CoA by pyruvate dehydrogenase. And so as like one of your colleagues said, obviously pyruvate dehydrogenase, it's a complex, it's a multi-enzyme complex. It is made up of 3 main enzymes, but you don't have to know them. You have to know it's a multi-enzyme complex. Okay, so for pyruvate dehydrogenase to be able to function, it needs 5 coenzymes. And if one of these coenzymes is missing, all right, if one of them is missing, it's not going to work anymore. So what are these 5 coenzymes? We have a mnemonic, which is the lovely coenzymes for nerds. Okay. So obviously V starts with a T. So the first one is thiamine pyrophosphate. Now, the second one, which is L, it's lovely. So L, so lipoic acid. Then coenzymes as in C, so it's CoA, our coenzyme A, and then F, which is FAD, and then N, which is NAD positive, right? So you do realize that they have like B1 and B5 and B2 and B3. The only one, so these are vitamins, vitamin B1, vitamin B5, vitamin B2, and vitamin B3. Okay. So how you have to know which one forms which one. Now, but we do have an exception, obviously, lipoic acid is not formed of any vitamins. Okay. So it's the only one with no vitamin, like it's not formed from any vitamin. Okay. So how do you memorize this? Like I'll try to simplify as much as possible. So as we know, thiamine pyrophosphate is the first one because T, so B1, vitamin B1. Lipoic acid is the only one with no vitamin synthesis, or is not made from any vitamins. Now for CoA and pantothen, like for CoA, you do have to just memorize that, I guess. It's B5. And you do have to know the other name, because this is extremely important to know the other names, because they do come at exams with the other name, and you like if you don't know them, you're not going to know it's FAD or NAD positive, right? For FAD, we know that Krebs cycle, as we said here. So wait, FADH2 forms 2 ATP, and NADH forms 3 ATP. So how you can memorize that is I mean, it's not really related, but just a way to memorize that is FAD, B2, because it forms 2 ATP, so B2, and riboflavin, because they have an F, so FAD. I mean, yeah, if you do want to memorize it that way, you can go ahead to do that. But if you just want to memorize that, that works too, but you have to know them, okay? NAD positive, obviously, 3 ATP, so also vitamin B3, and N, so niacin, so it starts with an N. So you can memorize that way. But yeah, again, you do have to know them. All right. Okay, so as I said, so let's say we have a disease. It's called the beriberi disease. Okay. Now, why is like beriberi? What happens in beriberi? So beriberi, what happens is we have thiamine deficiency, and we said if thiamine, or if one of these coenzymes is deficient, is PDH going to work or not?
It's not gonna work.
Okay, so it needs the 5 coenzymes to be able to work. Otherwise, it's not going to work. All right. So what happens if PDH does not work? So let's say PDH does not work. What is going to happen to, is pyruvate going to become acetyl-CoA anymore?
No.
Okay, so what's going to happen to pyruvate?
Will be accumulated in the blood.
Okay, so it's going to
The
increase.
pyruvate, it's going to be lactic?
Yeah, I'll explain that more, but you have to understand it's going to accumulate, right?
Yes.
Okay, so after it accumulates, 1 second. Okay, so after it accumulates, so pyruvate accumulates because this is no longer there. Okay, so after it accumulates, it's going to start to become lactate by lactate dehydrogenase. And this is also an important concept because lactate is acidic. Obviously, it's lactic acid, and it can cause lactic acidosis, which can cause weakness, which can cause also like this mainly affects the brain because the brain also depends on like the energy produced from TCA cycle. And if there is no TCA cycle, there is no energy produced. So as a result, it's going to form lactate, and lactic acidosis will be detrimental. Yeah, so this is also the same concept for pyruvate dehydrogenase deficiency, but the difference here is that pyruvate dehydrogenase deficiency can be congenital as well. So you can have pyruvate dehydrogenase deficiency due to beriberi, which is a lack of thymine, so it's not going to work. You can have it because it's congenital, so it's not there in the first place. And another point, or another third reason, which is extremely important, and it comes up in exams, is arsenic. Okay, so arsenic, it's a metal, it's a heavy metal. And this metal, what happens is, we said it inhibits pyruvate dehydrogenase. Now, we said, how do you inhibit pyruvate dehydrogenase? It's either you inhibit one of the coenzymes or it's congenital. So here it's not congenital. So you inhibit one of the coenzymes, okay? So in arsenic, what coenzyme does it inhibit? It inhibits lipoic acid. And lipoic acid, if there's no lipoic acid, then there is no pyruvate dehydrogenase, right? Or it cannot work, all right? So there is pyruvate dehydrogenase, but it cannot work. That's the difference between being congenital, that the congenital it has, like, it's not even there. But in beriberi or in arsenic, where there is arsenic, they inhibit it by inhibiting one of the coenzymes. Beriberi inhibits thiamine, or there is no thiamine, and arsenic inhibits lipoic acid. And so when there is no lipoic acid, obviously there's no PDH. Now, the clinical findings for this is extremely important. You have to know me lines, all right, which are these right here. These, all right. So me lines is very important because it also comes up in exams. You have to know me lines. You have to know the rest, but mainly focus on me lines. Me line, very important. Okay. So as of now, we discussed beriberi, which inhibits in which there is thiamine deficiency. So pyruvate dehydrogenase is not working. Second of all is arsenic, which is it inhibits lipoic acid. As a result, there is no pyruvate, or there is pyruvate dehydrogenase, but it does not work. And the third one, as I said, is deficiency and congenital, like it's not there, like it's not even there. So it's deficient. So that is why it's congenital. And as we said, it's converted to lactate. It causes problems for the brain because it cannot cause, or the brain needs the TCA cycle for the energy. Okay. So is everything fine till now? Because we're going to get into the actual TCA cycle. So is everything fine till now?
Yes.
Yes.
So
Okay.
I have a
Sorry?
beriberi and arsenic are not congenital, right?
Yeah.
So it's only deficiency.
Yeah.
Okay,
But
thank you.
arsenic, if you have if there's a metal, it causes that. Beriberi could be congenital because it is deficiency in thiamine. Like if you are deficient in thiamine, it could be congenital, or it could be genetic. Congenital means that it's like occurs at birth, right? So anything like deficiency could be congenital. But for example, arsenic is not congenital because arsenic, it's just a heavy metal, right? So it causes it inhibits lipoic acid and obviously inhibits PDH. Okay, so is that clear?
Yeah, thank you.
All right. Okay, so as I said, we'll be talking right now about the actual TCA cycle. So TCA cycle, which is also called the Krebs cycle or the citric acid cycle. These are the main three names of TCA cycle. Now, okay, so before that, we know what are oxidation reduction reactions. So whenever there is an oxidation or there is a transfer of one electron, so whenever let's assume there is something being oxidized, there has to be another thing which is being reduced, right? So oxidized is you're losing an electron, it's becoming more positive. And reduction, you're gaining an electron, so it's becoming more negative. And why am I talking about this right now? Because in the in the TCA cycle, what is being oxidized is acetyl-CoA, right? So we're oxidizing. One second. All right, so you're oxidizing acetyl-CoA. You're breaking acetyl-CoA down, you're oxidizing it, and what's happening is another thing is being reduced. And as we said, you're producing NADH and FADH2. So these are what's being reduced, and acetyl-CoA is being oxidized. Okay, so for the TCA cycle, we have four main reactions. Like it has a lot of reactions, but the four main ones that are going to focus on are the ones that produce the ones that produce the NADH and an FADH2. You have 3 NADH, 1 FADH2, and one, you can say it's ATP, but it's more like GTP. It's another form, but it's treated as an ATP. Okay, so so we have the four main reactions, which is the first one right over here. We'll be talking about each single one. This is the second one, the third one, and this is the fourth one. Okay, so the first reaction is isocitrate. Okay, so it's isocitrate forming. Second, okay, so it's citrate. Obviously, the citrate is isocitrate. Okay, so the first major reaction is isocitrate forming alpha ketoglutarate. So as I said, we're going to be tracing the ones that form NAD or NADH, FADH, or GTP or ATP. All right. So
To make, I just gonna ask you a question. To make citrate, don't we have to first add acetyl-CoA and oxalolactate?
Okay, yeah. So it's acetyl-CoA plus oxaloacetate gives
Will
citrate.
give us six carbon citrate, right?
Yeah, because acetyl-CoA is 2 carbons, and
Yes.
and oxaloacetate is 4 carbons. So it forms citrate, which is 6 carbons. But because like the ones in the doctor explained are the ones isocitrate, dehydrogenated. So I'm trying to focus on these because these are the main ones tested. Yeah, so like if you want to know it, mainly like it's these 4 reactions. So acetyl-CoA plus oxaloacetate forms citrate. They join together to form citrate. Now, citrate obviously forms an isocitrate. An isocitrate is basically it's an isomer. It's another form. There is no release of NADH. There is no release of CO2. Okay. Now, the first major reaction is isocitrate forms alpha ketoglutarate. Now, why is that a very important reaction? Does anyone know? There's 2 reasons it's important, this reaction here.
Forming NADH.
Okay, that is the first step. This is the first reason, but there's a major reason. The reason why it's extremely important.
Because you're producing CO2.
Sure, but no, that's not. Okay, so the main reason is that because it's the rate limiting step. So isocitrate to alpha ketoglutarate, this is the rate limiting step. So you know what rate limiting step is, right?
It's the
It
slowest step in
determines-
the reaction that's going to determine the speed of the whole reaction,
Okay,
whole
so
process.
all right, very good. So you have to understand what like for every single pathway you're going to take from glycolysis to TCA onward, you have to know what the rate limiting step is with its enzyme. So in this case, it's isocitrate forms alpha ketoglutarate. Okay, and we said that it forms NADH. So what is the enzyme going to be called? Isocitrate, which is the substrate, and dehydrogenase, because it forms NADH. At the same time, it releases CO2, right? So because it releases CO2 as well, so it's a decarboxylation. Okay, so but you have to know it's isocitrate dehydrogenase. It's the rate limiting step. It's the enzyme for the rate limiting step. Okay, so because it's why is that important to know the rate limiting step? The rate limiting step is important because it's regulated. It's the most regulated. So let's say we have a lot of ATP and a lot of NADH. What is going to happen to isocitrate dehydrogenase?
Negatively inhibited.
Okay.
It'll slow down.
All right, so it's going to inhibit isocitrate dehydrogenase because obviously we said the main point of Krebs cycle is to produce ATP and NADH, right? So if you have a lot of NADH or a lot of ATP, it's going to inhibit this step. Now we did talk about that in pyruvate dehydrogenase, but if you do get in the exam, if you do get isocitrate dehydrogenase and pyruvate dehydrogenase, you always go for the rate limiting step. Always, like if it's anti-regulation, ATP, NADH, you always go for the rate limiting step. If it's not there, sure, you can go for another step. But if there are 2 choices that work, you always go for the rate limiting step. Okay. Now, if we do have ATP and calcium, Now, as soon as you see a lot of ADP, or if you see an increase in ADP to ATP ratio, okay, you always assume that ATP is low. So you want to produce more ATP, right? So as a result, you want this reaction to proceed. You want Krebs cycle to occur, and you want this reaction to proceed because you have low ATP or high ADP, okay? And this is the first one, ADP. Now, the second one, which is calcium. Now, calcium, obviously, we know that calcium causes muscle contraction, right? And for muscle contraction to occur, you need a lot of ATP. So obviously, if you need a lot of ATP, it's going to stimulate this reaction. And that is why ADP and calcium, they both stimulate this reaction. Meanwhile, NADH and ATP, they inhibit this reaction, all right? So this reaction or this step, very important because it's the rate limiting step, okay? So is everything clear about this step?
Yes.
Yes, can I ask a question? So in the isocitrate, till it become alpha keto glutarate, what happens is that we release only one NADH and ATP. Do we also like lose a carbon? So like we lose from 6, it will become 5, right?
Yes. So so for isocitrate to alpha ketoglutarate, because it's a isocitrate dehydrogenase, you produce NADH, and it's also decarboxylated, so you produce CO2. So NADH is produced and you release CO2. So isocitrate become so it was initially 6 carbons, and now it's 5 carbons because one CO2 is released, right? So it becomes 5 right now. Okay, clear?
And do we also release ATP in this reaction, like between the isocitrate and the alpha keto glutarate?
No, it's only NADH and CO2. There's no ATP.
All right, thank you.
Okay. All right. So now for the next step, okay. So as we said, isocitrate forms alpha ketoglutarate by isocitrate dehydrogenase. Okay. So now we have alpha ketoglutarate. So alpha ketoglutarate is going to form succinyl-CoA. Now, how do you memorize again? So what is the substrate? It's alpha ketoglutarate. And we said the NADH forms NADH, right? So NADH is formed, so we can know that this is a dehydrogenase. So alpha ketoglutarate substrate plus a dehydrogenase forms NADH. Now, in the same way, it also releases CO2. So in the same way as the previous one, so isocitrate to alpha ketoglutarate gives is isocitrate dehydrogenase, so it gives NADH and it releases CO2. Alpha ketoglutarate to succinyl-CoA, so substrate alpha ketoglutarate, and it releases NADH, so alpha ketoglutarate dehydrogenase, and it also releases the CO2 because it's being decarboxylated. Okay, so for alpha ketoglutarate dehydrogenase, now you do remember pyruvate dehydrogenase. Remember the 5 coenzymes needed for pyruvate dehydrogenase?
Yeah,
Lipoic,
the B2 thiamine something, lipoic acid, FADH and ADH.
And
And there is
CaO.
one more.
Okay.
coenzyme.
The lovely
Coenzyme.
as an N, the T is thymine pyrophosphate made up of B1. Then lipoic acid, which is the lovely, so L, lipoic acid. Then coenzymes, it's CoA, and then coenzymes 4, so F, FAD, and then NERDs, it's NAD positive. Okay, so these coenzymes, which are used for pyruvate dehydrogenase or are needed for pyruvate dehydrogenase, are also needed for alpha ketoglutarate dehydrogenase, and this is presented right over here. So for alpha ketoglutarate dehydrogenase to work, it also needs the 5 coenzymes. You also, again, you have to know the other names: pantothenic acid, riboflavin, and niacin. Yeah, okay. So pyruvate dehydrogenase and alpha ketoglutarate, they both need these 2 enzymes. These 5 coenzymes. Okay. Now if you do realize, obviously calcium obviously simulates this step, and NADH inhibits it again as the previous step. But you have to understand, if you get calcium, for example, if you get ADP, and if you get, for example, alpha ketoglutarate dehydrogenase as well as isocitrate dehydrogenase, you're going for the rate limiting step. Okay, you're going for the rate limiting step. I hope this is clear. So does isocitrate dehydrogenase also require them? No, it's only alpha ketoglutarate and pyruvate dehydrogenase. Okay, it's only these 2 coenzymes or these 2 enzymes. They need the 5 coenzymes. Okay, so these 2 reactions, are they clear right now? So isocitrate forms alpha ketoglutarate by isocitrate dehydrogenase, and alpha ketoglutarate forms alpha ketoglutarate forms succinyl-CoA by alpha ketoglutarate dehydrogenase. Okay, so clear, right?
Yes.
Okay, do you have any questions till now?
No.
Okay. So again, so remember when we talked about beriberi, which is vitamin B1 deficiency?
Yes.
Okay. So we said that vitamin B1 or a thiamine deficiency, or in beriberi, what happens is there's thiamine deficiency. As a result, there is no thiamine pyrophosphate. And again, PDH will not work. And now in this case, because obviously alpha ketoglutarate also depends on the 5 coenzymes, it's also not going to work. So we have to correlate these two steps, alpha ketoglutarate as well as a pyruvate dehydrogenase. Beriberi obviously affects both. All right. And yeah, that's pretty much it for this slide. Any questions? If you do have any questions, because I don't want to be annoying. So if you do have any questions, just type in chat or you just can unmute, okay? All right. So isocitrate again forms alpha ketoglutarate, then succinyl-CoA. Now, the succinyl-CoA forms succinate. This is the third major step. Now, as I said, how do you memorize the enzyme? What is the substrate? Succinyl-CoA. So you put succinyl-CoA. Now, if you realize it's not dehydrogenase here, so there's no NADH or FADH2. Instead, it's synthetase, right? So instead, we have the formation of GTP, all right? And for that, obviously, because if you do realize we have a CoA here and no CoA here, so CoA is being released, okay? To become succinyl-CoA, to succinate, you're going to release CoA, and you're going to also form a GTP. And now this is important because, as I said, we're going to trace the steps for NADH, FADH2, and GTP. So this is the step for forming GTP. And now, because it's the first step, or because it's the step that forms GTP directly, we call this step a substrate level phosphorylation. Okay, so if you look here, so succinyl-CoA is being converted to succinate to produce GTP. So does anyone know what's meant by substrate level phosphorylation? So what is meant by phosphorylation or substrate level phosphorylation in this?
By releasing a phosphate group to create GTP or ATP. So like we're adding ATP to enzymes.
Okay, good. So you're adding a phosphate. So a phosphorylation is basically you're adding a phosphate. So to form GTP from GDP, you need a phosphate. So it's being phosphorylated, right?
Yes. Do we release the phosphate group from the succinyl coenzyme, succinyl CoA? Or like from where do we get this phosphate group?
So obviously, like for every GTP, there's GDP and a phosphate. So when it's broken down, so when GTP is broken down to GDP and there's a phosphate released, right? So by phosphorylation, there is like what we call a kinase, obviously. So it's going to join GDP to this phosphate, and it's going to phosphorylate it.
So it's like pairs, right? It's like pairs, they stay together, or they don't go like we don't sometimes. Like some reactions, we can like donate the phosphate group to the ATP or to GTP, and some reactions I don't think it requires that, right?
Sorry, some reactions. So if you want to form a GTP or ATP, you're going to need the phosphate because like it's 3 phosphates. GDP is 2 phosphates. So for it to form a third phosphate, you're going to need for it to form a third phosphate, obviously you're going to need an additional phosphate, right? So GDP 2 phosphate, GTP 3 phosphates. So to go from 2 to 3, you need another phosphate. So which is that? Make sense?
Yeah, it makes sense. I understand. Thank you.
You're welcome. Okay, so this is the third step, and this is the step is again substrate level phosphorylation. This is important, obviously, because again it gets tested in exams. They're gonna ask what a substrate level phosphorylation is, and obviously you do have to know phosphorylation GTP. Okay. For the third major step, okay, so I'm gonna go back here. Okay, so succinyl-CoA forms succinate. Now we have a succinate. All right, it gives fumarate. Okay, now this step, all right, so obviously we said how do you know the enzyme? What is the precursor or what is the substrate? It's a succinate. So it's succinate, not succinate. It's succinate. And obviously in this step, it's a dehydrogenase. So as I said, dehydrogenase, NADH or FADH2. The whole ones or the previous ones that we discussed, like dehydrogenases, they always produce NADH. However, for this step, it produces FADH2. And how you can memorize that is that it's getting converted succinate to fumarate. So F, right? So it produces FADH2. And another thing which is important is, remember, as I said, alpha ketoglutarate dehydrogenase, pyruvate dehydrogenase, they need 5 coenzymes. Now, succinate dehydrogenase, it only needs 1 coenzyme, which is FAD. And this is discussed here. It needs FAD. Okay, and FAD obviously comes from B2 or riboflavin. All right, this is a vitamin. Yeah. Now, so obviously, again, it requires FAD and it produces FADH2. Okay, to be able to function, it requires a coenzyme FAD and produces FADH2 as it's being converted to fumarate. So the enzyme is dehydrogenase because you're forming FADH2. And yeah, so this is the fourth step. Now, why is that important? Now, if you look right here, like in this slide, it says it's the only enzyme in the mitochondrial membrane. Now, we know that the TCA cycle occurs in the mitochondrial matrix. So because obviously it occurs in the mitochondrial matrix, like all the enzymes have to be in the mitochondrial matrix, except this enzyme, which is succinate dehydrogenase. Succinate dehydrogenase is not in the mitochondrial matrix, it's in the mitochondrial membrane. And I want you to keep this in mind. Okay, I want you to keep this in mind because as we go through the electron transport chain, it's gonna be there again because obviously the electron transport chain occurs in the mitochondrial membrane. Okay, so just keep this in mind, and we'll be talking more about that. Okay, all right. So this is mainly for the so this is the fourth step that you have to know. Obviously, if you do realize here, if you do really realize that there's another step which forms another NADH, which is malate to oxaloacetate, but so I don't think it's this like it wasn't discussed in your slides, so I don't think you have to know it. Just know like you just go over it in case it comes up in exam. Just know like it also forms an NADH. Okay, so obviously it's a dehydrogenase as well. The only one that forms FADH, dehydrogenase, which forms FADH is succinate because it forms fumarate. So F is fumarate. Okay. Okay, so as a result, we said, so this is the first NADH, this is the second NADH, and this is the third NADH. So we have 3 NADH and 1 FADH2 and 1 GTP. So 3 NADH, 1 FADH2, and 1 GTP. Okay, and this is for 1 pyruvate. That is why I said in the beginning, I asked the question is, why is it 2 pyruvate? Because 2 pyruvate enter the Krebs cycle. As a result, you're going to multiply the entire thing by 2. So technically, you have 6 NADH. So after 6 NADH, you're going to have 2 FADH2 and 2 GTP or 2 ATP. Okay. So I hope this is clear. Okay. So obviously, again, 3 NADH, you're going to multiply by 2. FADH2, you're going to multiply by 2, and GTP, you're going to multiply by 2. And these are important. Like the NADH and FADH2 are very important to reduce electron carriers. So they're important because they're also going to be used in the electron transport chain, and they're going to be used and they're going to be converted to ATP. Okay, I'll be discussing more in why is it 9 ATP and why is it 2 ATP in the electron transport chain. Okay. 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 Now, so are these steps clear for the electron transport chain? Like, do you have any questions about these steps?
Can you show me where's the FADH is being released? Which
Okay,
step?
yeah, it's not shown here, but I think there's another slide. But I'll show you. Okay, here. So succinate to fumarate, FADH2.
Can you explain from the fumarate to malate, like what happens? We add water to the fumarate through fumarase enzyme, and it becomes malate.
Yes. Okay. This is what happens. But again, as I said, like I don't want to discuss the entire thing because I think this is what doctors focused on, like the 4 main enzymes or the 4 main reactions that I talked about. But yeah, so because it's a fumarase, obviously, so you can add water and it becomes malate, right? So it's an ACE, so it's an enzyme. Then malate, obviously, it becomes oxaloacetate, reduces or forms NADH because
Through
it's a,
hydrogen.
yeah, it's a dehydrogenase, so it forms NADH. So the three main steps that forms NADH are the 3 dehydrogenases, except succinate dehydrogenase because it forms fumarate, so it's an F, right? And yeah, so again, before I move on to like electron transport chain and bioenergetics, is everything clear for electron transport chain or the TCA cycle?
Yes, it's clear.
Okay. And again, I want to like emphasize on the point that you have to know the steps for NADH, FADH2, and GTP. Okay, you have to know what the rate limiting step is or rate limiting enzyme is. Okay, so okay, so now what's said here is that TCA cycle is amphibolic. So does anyone know what amphibolic means or what it refers to? Okay, so amphibolic, so as we said that pyruvate or acetyl-CoA is being catabolized. Okay, so when you catabolize pyruvate, the point is you want to produce ATP, you want to produce NADH, you want to produce all of these for the electron transport chain. Okay, so it's being catabolized, it's being broken down. Okay, so now another thing which like the TCA can be used for is it can be used for anabolism. So what is meant by anabolism? It can be used for as in producing other things. So not only is it broken down, but it's also used to produce things. For example, we have a substrate in the TCA cycle. We have one intermediate, which is alpha ketoglutarate. Alpha ketoglutarate, it's used to form an amino acid. So that is why it's anabolic. So it's catabolic, obviously, because you're breaking down pyruvate and acetyl-CoA. But it's anabolic because it can, like the intermediates, can be used to form an amino acid, for example. Another intermediate, which is succinyl-CoA, can be used to form porphyrins or heme, which is like in the blood, right? So the point of this is that obviously it looks messy, right? But I'm assuming you only have to know the ones in the boxes, red boxes, because I remember we only got questioned about these. Because in MOL 2 and in later on, you're going to take gluconeogenesis, which I think it's the next PAL session. You're going to be talking about like how oxaloacetate can even be used to form glucose, and how like other amino acids they are used to form glucose. So yeah, it's going to become more clear and like in the next session. And I want you always to remember. I want you to remember how the TCA cycle is, like how it's related to all of that. I want you to remember how I want you to correlate everything. Okay, you can't study one thing then forget that thing and go on for another thing. You have to remember, you have to correlate things. Okay. So again, amphibolic because obviously it produces obviously it's being oxidized. Obviously, it's being oxidized. At the same time, it's being used for gluconeogenesis, which is in next lecture, for fatty acid synthesis, and for amino acid formation of amino acids, such as glutamate by alpha ketoglutarate. Again here, alpha ketoglutarate, and succinyl-CoA forms porphyrins and heme. Okay. So as we said, amphibolic, the term amphibolic is anabolic and catabolic, right? So now remember when I said that you're oxidizing acetyl-CoA, right? And remember I said the acetyl-CoA is made up of 4 carbons, or 2 carbons. The acetyl-CoA is made up of 2 carbons. Okay. So because if I want to form acetyl-CoA, so let's say it goes through this reaction. And again, this point is going to be clear in gluconeogenesis and other lectures. But I just want to give you just an introduction to what's going on. Okay, so obviously acetyl-CoA is made up of 2 carbons, and we said the 2 steps of dehydrogenase and as citrate dehydrogenase and alpha ketoglutarate dehydrogenase, they form NADH as well as CO2. Now, because the CO2 is released, so there's no CO2 more CO2s are released. And we said this has 2 carbons, like acetyl-CoA has 2 carbons, and 2 CO2s are being released. So with that being said, what happens is, if any of these, they cannot form acetyl-CoA anymore. Like I cannot use oxaloacetate to form acetyl-CoA And that is because this has 2 carbons and this does not have 2 carbons. I don't want to get into like details for this. You're going to take in gluconeogenesis and other lectures. Okay. So remember when I said that for example here, wait, let me show you. Remember when I said it can be used for glutamate, it can be used for gluconeogenesis or formation of glucose. It can be used for a lot of things, right? For example, porphyrins and heme. Now, we do have some reactions because you're losing this. You're losing malate, you're losing succinyl-CoA, you're losing alpha-ketoglutarate, you're losing oxaloacetate. So we have some reactions that maintain the levels of these. So you want to maintain the level of oxaloacetate, for example. You're using it for gluconeogenesis in the next lecture, but you want to maintain the levels of that. So we can use pyruvate directly to oxaloacetate, and this is by the enzyme pyruvate carboxylase. Okay, and this any reaction that is used to like replenish or to give back the intermediates that are lost. These intermediates or these reactions are called anaplerotic reactions. Okay, so they're called anaplerotic reactions. They're used to replenish what was lost, basically. Okay, so as said here, well, I'll show you. So an anaplerotic reaction, it's used to replenish the intermediates of the TCA cycle. And a very important anaplerotic reaction is pyruvate carboxylase, which basically you're forming oxaloacetate from pyruvate, directly from pyruvate. Okay, so this is just to replenish what was lost, because we said it forms obviously amino acids and other things, right?
Excuse me. Can you repeat like everything we took like at the start of electric transport chain? Like what's the start only after like the TCA cycle?
Repeat what?
I'm saying, can you repeat like everything after TCA cycle? If it's a lot, it's okay. You don't have to.
As in the amphibolic and anabolic and the
After the TCA cycle.
Okay. Yeah, fine. I'll agree with that. So we know. Okay, so you're gonna help me out. Okay. So we know the TCA cycle. It's used for oxidation. You're oxidizing acetyl-CoA, right?
Yes.
Okay, so you can say it's oxidizing acetyl-CoA or it's catabolizing acetyl-CoA. So they're
Yeah,
both the
we're
same.
breaking down.
Them down. Okay, you're breaking down
Yes.
acetyl-CoA. Okay, you understand that, right? That's the whole point.
Yes.
Okay, so now why do we not say the TCA is catabolic? Why do we say it's amphibolic? It's also anabolic. It's not only catabolic, it's also anabolic. So why is it
because we're also like building up, we're also creating ATPs, we're building ATPs, and we're also building an ADH. Not only that, we're also like, as you know, like we build like after malolate, we are also building oxalolactate too. So it's a cycle, basically. We start from we start from like we break down things and then we rebuild them from the start. It's a non-stop cycle, right?
Okay. You can understand it by that, but it also, remember I said the TCA cycle is very important for other metabolic pathways because it's also involved in forming amino acids. It's also involved in forming glucose by gluconeogenesis, which is the next past session. It's also involved in forming porphyrins or heme. So that is why it's like forming. I'm saying it's forming. So this is anabolic. So I said at the beginning, it's catabolic because it's breaking down glucose, but it also can be used to form glucose. It also can be used, like the intermediates can be diverted to form glucose. The intermediates can be used to form amino acids. Intermediates can be formed for porphyrin or heme. Okay, so this is why it's anabolic. That's why it's anabolic. Catabolic, because obviously that's the whole point of TCA is to break them down. So that's why it's not only catabolic, it's anabolic, and we refer to when like a cycle is both, we refer to it as amphibolic. Okay, makes sense?
Thank you. Yes.
All right. And as I said, what is an anaplerotic reaction? An anaplerotic reaction is basically
doesn't require oxygen.
No, anaplerotic reaction is because obviously, as I said, they're being used for other things, right? For example, alpha-ketoglutarate, glutamate, oxaloacetate is being used for glucose. So because you're losing these, so you want to replenish them because you don't want to You don't want to have nothing of this. You want to replenish them. So an anaplerotic reaction is a reaction that replenishes one of the intermediates. So for example, how can I replenish oxaloacetate? We can use pyruvate directly into oxaloacetate to replenish oxaloacetate, because obviously oxaloacetate is being used for gluconeogenesis. And yeah, so this reaction is an anaplerotic reaction, and it's by the enzyme pyruvate carboxylase. So when you study the gluconeogenesis, you come back to that, and you do realize that, okay, oxaloacetate, yeah, is being used for gluconeogenesis, so we have to replenish it by using this anaplerotic reaction, okay? So I know it's not going to be clear right now, but in the next lecture, it's going to become clear. When you get the bigger picture, it's going to become clear, okay? Everything is clear?
Yes, thank
Okay, so before we get into, I have like a couple of questions. Do you have any questions before we end the TCA cycle? If you do have any questions, just type in chat, or you can just unmute yourself. Okay, no questions? Okay. So all right, so the first question is, so who wants to answer this question? Can someone read it and answer it? Is anyone here? Hello?
Yes, I'll do it.
Okay, go for it.
24-year-old women present with diarrhea, dysphagia, and justice, sorry, whatever you call it, and white transverse line on a finger line, Mees lines. The patient is diagnosed with azenteric poisoning, to which of the following instance of the TC cycle? I know is that it inhibits the lipatic acid and
Inhibits lipoic acid, right?
yeah.
Okay. And lipoic acid is which enzyme needs the 5 coenzymes?
It's alpha ketolate dehydrogenase, and also we use it for alpha keto.
There's another one. What was it?
Yeah, pyruvate dehydrogenase
Okay,
also.
good. So because obviously pyruvate dehydrogenase is not the choices, it's alpha ketoglutarate. Okay.
Yes.
Karama, it's E. Okay, why is it E, Karama? Like, do you have any explanation for that? Okay, is it okay? If you don't understand that, let me know again. Okay. Okay, so the second question.
Can you re-explain it?
Okay, yeah, sure. So remember when we said arsenic? Remember when I emphasized an arsenic and the Me lines, right? Remember that?
Yes.
Okay. We said arsenic inhibits inhibits the lipoic acid, right? So wait, I'll show you. Okay, inhibits lipoic acid. So slide 21. Okay. So actually, wait, the Me lines. Okay, slide 9. Okay, so we said arsenic inhibits lipoic acid, right?
Yes.
Okay, so because it inhibits lipoic acid is one of the 5 coenzymes. It's one of the 5 major coenzymes. And the 5 major coenzymes are used for pyruvate dehydrogenase and what is the other one?
I guess alpha keto glutarate.
Alpha ketoglutarate, yes. So alpha ketoglutarate and pyruvate dehydrogenase. So because you inhibit lipoic acid, this is going to be inhibited as well. So pyruvate dehydrogenase and alpha ketoglutarate. Okay, makes sense?
Okay, thank you.
You're welcome. Okay. Now the next question. I know I showed the answer, but it was a mistake. All right, anyway. So who wants to read that question? But you have to explain it because I know I showed the answer, so you're gonna have to explain it. Okay. Anyone willing to try?
Is it D?
Okay, yeah, it is D. But why is it D?
Well, this step requires FAD. the coenzyme FAD, which comes from riboflavin.
All right,
I think
sounds
it's
good.
B4 or B5. I don't remember.
Riboflavin, it's so okay. So riboflavin is FAD, right?
Yeah,
Okay, and we know in the like electron transport chain, FAD is used to produce how many ATPs?
2,
Okay, so you can memorize that as 2 and B2.
B2.
So, yeah,
Thank you.
you're welcome. Okay, so this is the second question. If it's not clear, just let me know in chat again. Okay, third question. Okay, so who wants to do it? Okay, anyone wants to do it? Okay, go for
D,
it. Okay, yeah, it is D. So why is it D?
because it's a synthetase, and we're basically synthesizing GTP from
Okay,
its
so yeah,
name,
so
and it's
yeah,
also in like the process of turning succinate into turning succeedal or something like that to succinate.
it's succinyl-CoA to succinate. So
Yeah.
you're using it by succinyl-CoA into succinyl-CoA synthetase, and it's also the substrate level phosphorylation GTP to GDP. Okay, is that clear for everyone? Are these questions clear? Because I'm gonna
Can you
get
share later?
sorry.
Can you share the questions with us later?
Yeah, sure. Okay. Okay, so I hope everything is clear for the TCA cycle. I hope I know it gets easier like the first time you study, it gets easier like if you understand how to like correlate the step with the actual enzyme and its name, it becomes really easy. Okay. So now we'll get into the electron transport chain, but for some reason like they explained bioenergetics at the beginning of the lesson. So I'll do that as well. Okay, so obviously we have what we call thermodynamics as in heat and how heat changes basically. I'll just go over it like briefly over it because the main thing is electron transport chain. Okay, so there are 2 laws of thermodynamics. The first law, it just emphasizes on the point that you cannot create energy or you cannot destroy energy. The entire amount of energy is always like the same. So you cannot destroy it, you cannot create it. It's just when you find a different type of energy, it's because it's being transported. It's being like it changes form. The form changes, but it's not destroyed or it's not created. Okay. Now, this is the first step. The second step is or the second law, it's basically what we call it's basically based on entropy or the disorder of the universe, like how the actual particles of the energy, how distributed they are. Okay, so what it says is any change or physical or chemical change always has an increase in disorder of the universe, in disorder or entropy of the universe. Okay, so disorder where like the degree of order is also called the entropy. Okay, now for like this is an important point as well, because for living organisms, again, this also applies to it. There also must be an increase in entropy. Okay, so just know this point that for living organism there is an increase in entropy every time there is a change in the form of energy. Okay, so so we have what we call the free energy change or the free energy or Gibbs free energy. Okay, so the free energy is the energy the energy that is being used, or the energy it's either being used or the energy that we need for a reaction to occur. So remember here in catabolic and anabolic. So just go here. Okay, so for anabolic reaction and catabolic reactions. So catabolic reactions, you're taking something and you're breaking it down. So when you break something down, do you need ATP or are you going to release ATP? Because obviously there are bonds and there are like ATP within these bonds. So when you break the bonds, you're going to release the ATP. Okay, so this is why it's a catabolic reaction, and this is also referred to as an exergonic reaction because you're releasing ATP. Okay, but exergonic is more of heat. All right, so anabolic reaction, it's the opposite. You have like simple things and you're trying to combine them to form a more complicated thing. And for that to happen, you're going to need energy. So because obviously, like, you have two small things, you want to form more, you're going to need the energy. If you have a large thing, you break it down, you're going to release the energy within these bonds of the larger thing. Okay, so this is just to understand what's going on. So as I said, free energy, it's the energy or the like the energy that we either need or the energy that is being released. It depends on the type of reaction. Okay, we'll be discussing it. So if you want to find the free energy change, it's basically any word to change means it's the energy of the product minus the energy of the reactant. So you're going to subtract them and you're going to find the free energy change. Okay. Now, keep this in mind because like keep the free energy or Gibbs energy in mind. It's the major thing that like it's the most important concept here. Okay. So obviously you can also find Gibbs energy. There is another reaction or there is another like equation which it's called the Gibbs-Helmholtz equation. And now this equation basically relates the Gibbs energy or a change in free energy with enthalpy as well as entropy. So what is enthalpy? Enthalpy is it's energy, but it's in the form of heat. Okay. So when energy is released as heat, this is what we call enthalpy. It could be absorbed, it could be released. Now, what about entropy? Entropy, as I said, it's how random, like how random or yeah, it's how random or how diverse it is. Okay. So the point is when there's a negative or a positive delta G, okay. So this means here there's a negative, let's say there was a positive delta G. Okay. So this means the energy of the product is more than the energy of the reactants because obviously this is higher and this is higher than this. So obviously this is going to be positive. So because it's positive, so for the reactant to become a product, it needs energy. So if it needs energy, you can say that this is an endergonic reaction here. Okay, it needs energy. It's non-spontaneous because it needs the energy. However, an exergonic reaction, it's spontaneous because you already have the energy. So let's say you have 10 volts or whatever the joules, I don't know what the is. Yeah, kilojoules per mole. Okay, so let's say you have like 10 and this is like the reactants have 10 energy, like form of energy, and products have 5. So it's easy to go from 10 to 5. However, if there's like the opposite way around, so it's 5 and you want to form and you want to go to 10, you obviously have to gain the energy. So because you have to gain the energy, it's endergonic and it's non-spontaneous. Okay, so exergonic has a negative because how you can think of it, it's exergonic. So you're releasing the energy because you already have enough. Endergonic, you're going to need the energy. You're going to need the energy to form like the product. Okay. So this is obviously spontaneous because it forms without like adding the energy. And here it's non-spontaneous because you need energy to form it. Okay. And as I said, these are the most important points, the delta G and how it's related to endergonic and exergonic reactions. Okay. Yeah. So the point is, when I say that, obviously we said it could either be negative or it could be positive delta G, right? So when it's either negative or positive, the point of the reaction is that you want to reach equilibrium. So when I say equilibrium, I'm talking about you want to be like the reaction wants to become stable. Okay, so reaction wants to become stable, like there is no change over time. So like the forward reaction is equal to the backward reaction or is equal to the reverse reaction. And what that means is delta G is going to become 0 because when delta G is 0, these reactions are equal. So this forms that and that forms that as well. Okay. However, when it's obviously negative, only the reactants can form the products, but they need energy. If it's positive, if it's negative, they're going to release energy. If it's positive, they're going to need the energy. All right. So they always tend to form. So when I say it's spontaneous, they always tend to form. They always tend to go to equilibrium. That is the whole point of all reactions. You always want to get to equilibrium and you want to be you want the reaction to become stable. Okay. Yeah. So all right. So as we said, we have what we call the delta G and the delta G naught or whatever you want to call that. So the delta G, it's we know if you want to measure delta G, you want to take into consideration the function of reactant and the product concentrations and of the temperature. Now this is what is the difference between these is that this one, All right, this one, it's the standard free energy change. So what that means is you already have the temperature, you already have the pressure, and you already have the concentration of the reactants and the products. So this is the standard one. However, the actual one, obviously it differs from one reaction to another. This is why it's called the actual one, because of it's the delta G of the actual reaction. Okay, it's not the standard. All right. So obviously these are the like it occurs at 298 Kelvin and 1 atmosphere pressure with concentrations of reactant products of 1 molarity, right? So that's why you have to know, like, these have known values, these do not have known values, and this is why it's the actual one. Okay. Okay. So as we said, is standard free energy change. Obviously, standard temperature is this, as 298 Kelvin or 25 degrees Celsius. Standard temperature is 1 atmosphere pressure. And the initial concentration of reactants and product is 1 molarity. So these are the defined reference conditions. Okay. Now, we do have another way, or we do have a way where we can correlate delta G, like the standard free energy change, with equilibrium. Okay. So we know when there is equilibrium. So let's take this into consideration first. Okay. So when there is equilibrium, concentration of A, okay, and concentration of B, obviously, because they're in equilibrium, so they're both going towards each other, right? So but because they're both going towards each other, they're at equilibrium. So what I can do is I can remove like concentration of B over concentration of A, and I can replace it with equilibrium because they're both at equilibrium. Okay, but to consider that both at equilibrium, remember that I said the delta G has to be 0. When if they are at equilibrium, delta G at 0. So I can only replace this by equilibrium only if delta G is 0. If delta G is not 0, you cannot replace it. Okay, you cannot replace it, but because it's not at equilibrium. So if it's at equilibrium, you can replace that by 0. And if you replace that by 0, obviously you take this to the other side and it's going to become negative, and you replace that by equilibrium. Now, obviously the R is basically the ideal, it's a constant, it's an ideal gas constant. The T is the temperature in Kelvin, and it's not in degrees Celsius. This is a common trick, like it gives you in degrees Celsius. And then you have to convert it to Kelvin, and this is the equilibrium. Obviously the K equilibrium, how do you calculate it? It's B over A. Okay, so does that make sense for everything here? I was just trying.
Can you please repeat the last slide?
This one?
Yes, please.
Okay, so as we said, delta G. So let's say we talked about delta G, and delta G, like the standard free energy change. Okay, this and this, the actual, which is that, and the standard. Okay, so we have an equation that can like link them both. Okay, so it's delta G equal delta G like Nadh and plus RT and ln, which is a logarithm. Like concentration of B over concentration of A. Okay, so at equilibrium, we said, remember what happens at equilibrium? We said the delta G is going to become 0, right? That is how you know it's at equilibrium. If it's negative, if it's positive, they're not at equilibrium. To become at equilibrium, delta G has to be 0. So you replace this by 0. All right. Okay, so after you replace this by 0, we said we're at equilibrium. So at equilibrium, I can replace the concentration of B over concentration of A with a constant of an equilibrium constant. And why is that? Because they're both going towards each other. At equilibrium, there's no change. There's no net change. This is forming that, A is forming B, and B is forming A. Okay. So as a result, I can replace B over A with K equilibrium. So you're going to form delta G, the standard, okay, it's going to become negative RT because obviously this becomes 0, right? So you're going to take this part over here to the other side. I mean, this is just math at that point, but this is how this reaction is brought up. So it becomes negative, and this is why there is a negative sign right over here. For the R, it's an ideal gas constant, which is right over here. It's always the same value. For T, it's Kelvin, it's temperature, but it's in the form of Kelvin. It's not in the form, it's not in degrees Celsius, it's in Kelvin. And as we said, K equilibrium is there because we're at equilibrium. Okay, so the only way we can use this if it's at equilibrium. If it's not at equilibrium, you're gonna have to use this. Okay, so is that clear?
Yes, thank you.
All right. Okay, so as we said, we already talked about that as an exergonic and intergonic reactions. Obviously, ADP, you want to form ATP, so you're going to need energy, right? You're going to need energy. And where is this energy brought from? So we have other exergonic reactions, they release the energy, and they give this energy for the formation of ATP. As ATP becomes broken down to ADP and phosphate, it releases energy. And this is the energy used for intergonic reactions. So it's like ATP and to broken down is an exergonic reaction, or it's being catabolized. So this energy which is being used is for intergonic reactions. So it's not an so this right here is not an intergonic reaction, it's exergonic, but the energy released, because we said you cannot destroy the energy. So where's the energy going to? It's going to other reactions, and these other reactions are going, like, they are intergonic reactions because they need that energy. And the same concept goes to here. ADP, you're gonna give you want to combine it with phosphate to form ATP, and obviously, as we said, this is not an exergonic reaction, but because you need the energy from other exergonic reactions to form this reaction. Okay, I know it's kind of like this slide is kind of confusing, but I hope this is clear. And we already talked about that. Okay, so everything is clear for bioenergetics because we're going into the electron transport chain. So do you have any other questions for either bioenergetics or the citric acid cycle or a Krebs cycle? Okay, no questions. All right. So now I'm assuming bioenergetics is like it was introduced because like it wants you to understand which reactions are spontaneous and which reactions are not. You're gonna understand that obviously later lectures as well. Okay, anyways. So now we discussed, obviously this is glycolysis, it was already discussed. We discussed this part, and now we already formed. We said, I want you to link everything together. We said we formed 3 NADH, 1 FADH2, and 1 ATP or GTP. Okay, so these NADH and FADH2, they will go to the electron transport chain. And in the electron transport chain, they're going to start to produce ATP. And I'll be talking about how that happens. So just to understand, 1 NADH, like for every single NADH, it forms 3 ATP. And for every single FADH2, it forms 2 ATP. Okay, so NADH is 3 ATP and FADH2 is 2 ATP. Okay. So so for the electron transport chain, first things first, we said that the TCA cycle obviously occurs in the mitochondrial matrix. So the like right over here, this side over here. Okay. However, the electron transport chain occurs at the membrane of the mitochondria. Specifically at the inner membrane of the mitochondria, which is called the cristae, or the folded inner membrane, okay, which is also called the cristae. All right. So what happens is, obviously, we said that NADH and FADH2, they go to the electron transport chain and they start to produce ATP. So what happens is these NADH and FADH2, they're in the reduced form. Okay, they are reduced from the TCA cycle. So when they are reduced, after they go to the electron transport chain, what's going to happen is they will donate an electron. So now what you have to understand is NADH differs from FADH2 in one step. Okay, so let's talk about NADH first. Okay, so NADH, what happens is obviously it's reduced. It's going to donate an electron to NADH to complex number 1. Complex number 1 is the first component of the electron transport chain. Okay, so what happens is it donates an electron and it becomes oxidized. So when it donates an electron, here, okay, to complex number 1. Complex number 1 is also called NADH dehydrogenase. So NADH dehydrogenase, because they only accept electrons from NADH. Now, if we're talking about FADH2, it skips FADH2. If you realize, it doesn't go to complex number 1. It goes to complex number 2 instead. So it skips complex number 1. It goes to complex number 2. Okay. And then, so what happens is NADH. So NADH is a complex number 1. So it has a lot of electrons. It donates an electron to complex number 1. Now, when it donates an electrons, this complex is going to become charged. So it's going to become charged, and what it's going to do is that it's going to pump protons to this side. So this is the mitochondrial matrix. It's going to start to pump protons to the intermembrane space. Okay, so this is the first step. It's going to pump protons into the intermembrane space. Okay, so this is complex number 1. After complex number 1, this is for NADH. Okay, not FADH2. You have to understand the difference. So what happens to like the electrons is that the electron from NADH, it goes to coenzyme Q. Coenzyme Q is also called ubiquitin. So coenzyme Q is also called ubiquinone. All right. So ubiquinone. And all right, so it's also called ubiquinone. All right. So after coenzyme Q, it goes to like the third complex, complex number 3, and complex number 3 pumps protons. Then after complex number 3, it goes to cytochrome C, and then from cytochrome C into the complex number 4. Complex number 4 pumps protons. So the 3 main things that pump protons are complex number 1, 3, and 4. Coenzyme Q and cytochrome C, they do not pump protons, and complex number 2, leave complex number 2 alone for now. So coenzyme Q and cytochrome C, they do not pump protons. It's only 1, 3, and 4. Okay, they pump protons, and the point of that is that they want to create a proton, like a gradient. Okay, proton gradient. Okay, so now, so as a result, so we know that complex for NADH, complex 2 is not involved. This is why in this slide it says that for NADH, okay, for NADH, it's only complex 1. Then it goes to coenzyme Q. Then from coenzyme Q, it goes to cytochrome C. It pumps protons. And this pumps protons as well. Then cytochrome C goes to complex 3 goes to cytochrome C, which is mobile and water soluble. All right. So coenzyme Q and cytochrome C are both mobile. All right. So it doesn't pump protons. Then from cytochrome C, it goes to complex number 4. And complex number 4 is also known as cytochrome oxidase. You have to know those other names. You have to know the other names. For every single one, you have to know the other names. Okay. So why is it known as cytochrome oxidase? Because it takes this electron. So remember here, we said that electron is at this point right here. So it takes this electron and it gives it to an oxygen. Now that is why oxygen is very important at this step. Oxygen is the final electron acceptor, because obviously electron goes from complex number 1, coenzyme Q, complex number 3, cytochrome C, complex number 4, and then where is it going to go? Here comes the oxygen. It binds to it, or it accepts this electron and it forms two water molecules. So it binds to so oxygen binds or combines or accepts the electron to form water. Okay, so now this is for NADH. Now FADH2 is a different story. It's just slightly different in that it doesn't go to cytochrome, it doesn't go to complex 1. Instead, it goes to complex number 2. It goes to complex number 2. And then it follows the same path as the NADH, the electron from NADH. So FADH2 donates an electron to complex number 2, and after it donates an electron to complex number 2, it's going to go to cytochrome or coenzyme Q. So similar to NADH, it goes to coenzyme Q, and then another point which is important. We said for NADH, complex number 1, 3, 4, the pump protons. Now for FADH. For FADH2, when it donates an electron to complex number 2, it does not pump protons. So the only one that pump protons are 1, 3, and 4 for the entire thing, even for NADH. But NADH obviously doesn't have, it doesn't involve 1. It only involves 2, coenzyme Q, 3, cytochrome C, and 4. Okay. Now, so as we said, this does not pump protons, and this is important to understand because obviously it gets tested. So pump protons, pump protons, pump protons, does not pump protons, do not pump protons. Okay. So what happens is, now I want you to recognize something. So if you recognize here, what is complex number 2 also called? It's called succinate dehydrogenase. Now, succinate dehydrogenase, remember, where is it also found? It's also found in the TCA cycle. So so I'll show you, wait. Okay, so it's also found in the TCA cycle. Remember succinate dehydrogenase when I said I want you to remember that because it's the only enzyme in the mitochondrial membrane. It's the complex number 2 for FADH2 because it's complex number 2 to form FADH2, like succinate to fumarate, and it's also the enzyme that accepts the electron from FeDH2. That makes sense. So what you have to understand, so that is why I want you to correlate things together, because complex number 2 is also involved. So it's not only involved in TCA, it's also involved in the ATC and the, like electron transport chain. And that is why it's the only enzyme which is found among the TCA cycled enzymes. It's the only enzyme which is found in the mitochondrial membrane, because this is where electron transport chain occurs. Okay. So as you can see, it says here, obviously succinate dehydrogenase catalyzes oxidation of succinate to fumarate, and it also takes the electrons. It takes the electrons from FeDH2 to coenzyme Q. So because it's complex number 2, so succinate dehydrogenase is also complex number 2. So complex number 2, which is that right over here, it's that, sorry. So it's not that, it's that. So this is complex number 2. FeDH2 donates the electron to complex number 2. It goes to coenzyme Q. Complex number 3, cytochrome C, and complex number 4. Okay, so I hope this makes sense. Yeah, I hope that makes sense. And again, complex number 2, no protons are pumped at complex number 2, only 1, 3, and 4. Okay, and yeah, so this is basically what I talked about. And now, so we said, okay, so like the electron, we said we pump protons, 1, 3, and 4 pump protons, and the electron is being transported like through the enzymes, and then it's going to become accepted by O2 to form H2O or water. So as a result, what's going to happen? We said we have a lot of protons. You're going to pump your protons here, and these protons, they're going to form a gradient. Okay, they're going to form a concentration gradient, and this concentration. So what happens here? This concentration gradient. Like, because obviously we know like they tend to go from high to low, right? So we have an enzyme that helps for it to transport the protons. These protons are going to go back into the like the mitochondrial matrix. And the point of that is that, remember when I said, if you want to form ATP from ADP and phosphate, you're going to need energy. And this energy comes in the form of these protons. These protons, they help to cause this formation of ATP. Okay, so this is how it's correlated. So first, obviously they pump protons here, and this proton is going to create a gradient, and this gradient is like it's going to be pumped back by ATP synthase to stimulate the formation of ATP, because you need the energy, right? It comes in the form of H positive, but you need the energy of ATP. So this energy in the form of H positive, it's like it stimulates the formation of energy in the form of ATP. Okay, so now this part over here, specifically right over here, is what we call oxidative phosphorylation. Oxidative phosphorylation, it's that part over here, and the electron transport chain, it's where the electron is being transported. It's a chain of where the electrons is being transported. So this chain, it's that part over here, which are the enzymes I talked about, and the ATP synthase is only oxidative phosphorylation. Okay, so I hope that makes sense. Chemiosmosis, it's what I talked about. So it's basically you're using the energy by H positive gradient or the proton gradient to form the energy of ATP, and this is what we call chemiosmosis. Okay, chemiosmosis, right? So obviously it's an ATP synthase that pumps it at the last part. I already discussed that. And yeah, okay. So that's again the same exact point. You form a proton gradient, it's being pumped back by ATP synthase, because obviously we said that you need to form ATP, and this ATP is being formed by another exergonic energy, which is coming from H positive, right? And this process is what we call oxidative phosphorylation. Okay, so I already discussed that 1 NADH forms 3 ATP, and 1 FADH2 forms 2 ATP. Okay, so is everything clear for the electron transport chain? Because yeah, so we're done with that. We have just the inhibitors and the uncouplers. So do you have any questions regarding the components and like how the electrons are passed through, how ATP is synthesized? Do you have any questions? Okay, so the complexes that do not pump proton, so it's complex number 2 and coenzyme Q and cytochrome C. The only ones that do pump is 1, 3, and 4. Okay, and why is that important? Because FADH2, it only goes to the 2, and it doesn't go to 1. So it goes to complex 2, then coenzyme Q, then 3, then cytochrome C, and then 4. Okay. Yeah, that is the answer to your question. Like 2 coenzyme Q and cytochrome C, they do not pump protons. Okay. Okay, any other questions? Is everything clear? Okay. Okay, so now we'll be talking about something which is extremely important as well, which are the inhibitors. And why is that important? I'm not just saying it's important because I think it's important. It's important because it's tested. It's tested a lot of times. Okay. So obviously, electron transport chain, oxidative phosphorylation. Oxidative phosphorylation is only the part where ATP synthase is involved, and like ETC is where the electron is being transported through the complexes. Okay. Now let's say any of these steps. Let's say it was inhibited. So if you inhibit one step of these, the entire thing will be inhibited, because obviously, like let's say you inhibit complex 1. If you inhibit complex 1, electron cannot go to coenzyme Q, then complex number 3 and cytochrome C and 4, right? So this is why oxidation of NADH is reduced, because you cannot, like, the whole thing is stopped. Oxidation of NADH, as in you cannot use NADH to form ATP. So that is why it decreases. And now this is important as well. The second thing which is important, obviously, oxygen consumption decreases. Remember where we said oxygen is used? Oxygen is used at the, like, just right the end, after complex number 4. Complex number 4, what happens is, like it donates the electron to an oxygen, and as a result, it forms water. So if you don't have the electron in the first place, like if the reaction is inhibited in the first place, there is no oxygen in the first place. So that is why there is oxygen, but you're not using the oxygen. Okay, because there's no electron to accept, so the oxygen is not being used. That is why decreased oxygen usage or consumption. Okay, and obviously decreased ATP synthesis, because that's the whole point of ETC or electron transport chain, is to produce ATP. And if there's, like, if it's inhibited, if it's the process is inhibited, you cannot produce any more ATP. Okay, so you have to know these. Obviously, I mean, this is self-explanatory, but it gets a little bit confusing in the uncouplers, but just know that. Okay, so as I said, we have inhibitors, and very important inhibitors that you have to know. You have to know what they are, and you have to know what they inhibit. So for the first one, for complex number 1, which is also called NADH dehydrogenase, because it accepts NADH only. Anyways, so first one, we have the rotenone and barbiturates. Okay, these two inhibitors, they're extremely important, and they inhibit complex number 1. So they inhibit complex number 1. This is the first thing that you have to know. Now, another thing which are cyanide and carbon monoxide. Now you can memorize that as in C and C. They're together, and they inhibit complex number 4. Okay, they inhibit complex number 4. I'll be talking about them more. And then oligomycin. Oligomycin inhibits ATP synthase. It prevents, like, the proton channel. We have ATP synthase. It has a channel to allow the protons to pass through to the mitochondrial matrix. It inhibits this channel. Okay, so rotenone and barbiturates for complex number 1, cyanide and carbon monoxide for complex number 4, and oligomycin for ATP synthase. Now, the most important thing, obviously, again, rotenone, complex number 1, cyanide, carbon monoxide, complex number 4, and oligomycin ATP synthase. Okay. Now, the most important thing that we want to talk about are cyanide poisoning and carbon monoxide. Okay, and carbon monoxide. Now, for cyanide poisoning, what happens is it binds irreversibly to complex number 4. And what is meant by irreversibly is it binds tightly to complex number 4. So when it binds tightly to complex number 4, it prevents the electron from, like, it prevents, we said, what is the main job of complex number 4? The main job of complex number 4 is that it gives an electron to the oxygen. It gives an electron so that the oxygen can accept that electron and forms water. Okay, so if the complex number 4 is inhibited, the electron will not be transferred to oxygen. Okay, the electron will not be transferred to oxygen, and this is how basically you're not using oxygen, right? And obviously this happens in tissue hypoxia. What is tissue hypoxia? Hypoxia as in there is no oxygen. When there is no oxygen, there's nothing to accept the electron. Okay, so this is how you have to think of it. What is the function of complex number 4? To transfer the electron to for the oxygen to accept it. If cyanide poisoning binds to it, it prevents this transfer. Or if there is no oxygen, you cannot bind to, like the oxygen cannot bind to the electron because there's no oxygen, right? So this is the first point about cyanide poisoning. Now, carbon monoxide. Carbon monoxide, again, it inhibits complex number 4. However, it's a bit different. So what happens is, obviously, it binds to complex number 4, but it's not, we said it like cyanide, it binds irreversibly. So it binds tightly. But carbon monoxide, it binds less tightly. However, what carbon monoxide does, we know that hemoglobin carries oxygen, okay? So what carbon monoxide does is that it binds to the hemoglobin, and when it binds to the hemoglobin, it replaces this oxygen. So the oxygen is no longer carried by the hemoglobin. So what you have to know about carbon monoxide and cyanide is that they inhibit complex number 4. If you inhibit complex number 4, the electron will not be transported. Like it will not be, like you can, like there's the oxygen is there, but you cannot accept the electron because you cannot transport it because it's inhibited by cyanide and carbon monoxide. Okay. Okay, so as I said, these are the inhibitors: rotenone, carbon cyanide, rotenone inhibits like the first one, which is NADH dehydrogenase or complex number 1. Cyanide and carbon monoxide inhibit complex number 4, and oligomycin inhibits the last step, which is part of the oxidative phosphorylation, which is ATP synthase. Right? These are the inhibitors. Is this clear? Is everything about this part clear, like the inhibitors?
Yes.
Okay. Okay. So as I said, we have something called inhibitor and we have something called the uncouplers. Okay. So what the uncouplers do, these are the uncouplers. So what uncouplers do is that we said, obviously the complexes, they pump the protons to form a proton gradient. Okay. So what uncouplers do is that they make the membrane leaky. And when this membrane becomes leaky, the protons are going to go back. And when they go back, there is no longer going to become a gradient. Okay. So technically, it does decrease ATP synthesis because the protons are going back and they're not being used by ATP synthase. Okay. However, the difference between uncouplers and inhibitors is that uncouplers, they increase oxidation of NADH. Remember that inhibitors, they do not increase it. Inhibitors, they decrease it. They decrease the oxygen consumption and they decrease ATP synthesis. They both decrease ATP synthesis, but this increases oxidation of NADH and increases oxygen consumption. And why is that? Because when there is no electron or when like there is no proton gradient, like the proton gradient is lost, right? So electron transport chain, it's going to keep on forming, like it's going to keep on going because there's no gradient. You want to form a gradient. So it's going to keep on going. And for it to keep on going, it's gonna use NADH. So it's going to oxidize NADH. That's the first point. And the second point is going to be using oxygen consumption. It's going to be using oxygen to like accept these electrons. However, why is ATP synthesis not used, or why is there no ATP synthesis? Because ATP synthase is not being able to pump the protons because obviously the protons are already being leaked. Okay, so instead, what's going to happen is energy is being released as heat. Okay, so instead of forming ATP, energy is being released as heat. So you have to know these. When there's a lot of oxygen used, or when there's a lot of oxidation of NADH, but no ATP synthesis, this is an uncoupler. However, when they all decrease, this is an inhibitor. Okay, so here it does form energy, but it's in the release, it's in the form of heat, and that is why, for example, newborns have what we call the brown adipose tissue. The brown adipose tissue in the mitochondria, they have an enzyme, or they have an uncoupler called the UCP1, which is one of the uncouplers here. You have to know the uncouplers, which is 2,4-dinitrophenol, or 2,4-DNP. Aspirin is also an uncoupler, and UCP, or a thermogenin, which I talked about, and I'm going to talk about again. So again, newborns have brown adipose tissue, and this brown adipose tissue, the mitochondria, they have an uncoupler, which is called UCP1, which is this, or thermogenin. And what that does, why is it important for newborns? Because it keeps them warm. And the point of keeping them warm, and how does it keep them warm? Because obviously it produces energy in the form of heat. It does not produce energy in the form of ATP, it produces energy in the form of heat, and it keeps them warm. Okay, so you have to know these three uncouplers, 2,4-DNP, aspirin, and UCP, or thermogenin. Okay, to maintain the body temperature, as we said. Okay, All right. So obviously UCP it functions as other uncouplers. So what it does is that it makes the membrane mitochondrial inner mitochondrial membrane leaky, and as a result, the protons they go like there is no concentration gradient anymore. Okay. Okay. Can you please repeat the uncoupler slide? Yeah. Okay, sure. So uncouplers, what they do is the main job of uncouplers is that they make the mitochondrial membrane leaky. So when the mitochondrial membrane becomes leaky, we said the point, obviously we have a lot of complexes, they pump protons. Okay, but it's leaky. So what's going to happen here is that the protons, they're going back to the mitochondrial matrix because it's being leaky. Now, obviously, we do have the protons. We want them to go back at the end, but we want them by ATP synthase to use them for formation of ATP. Okay. However, here they're not being used in that way. They're being used as in before that in the electron transport chain. And what happens is they're being leaked into the mitochondrial matrix, and as a result, because of their leakiness, as a result, there's no gradient anymore. And because there's no gradient anymore. So because there is no gradient anymore, like electron transportation is going to keep on going. It's going to keep on going. The rate is going to increase because nothing is going to stop it. Okay, because obviously they're being pumped back, and the point of it is you want to pump it outside, but they're being pumped back, so it's going to keep on going. And when it keeps on going, it has to use NADH and it has to use oxygen. That is why NADH oxidation increases and oxygen increases. Okay, oxygen consumption increases, like the use of oxygen increases. Okay, however, obviously, because it's not being used by ATP synthase, you're going to decrease ATP synthesis. So in uncouplers and inhibitors, they both decrease ATP synthesis, but these 2 points are the different points. Okay. Obviously, as I said, because they're not being used as for ATP synthesis, instead they're being used for energy. They're being like the energy is being used for heat, and that is why in newborns have one uncoupler, which is called the UCP or thermogenin in brown adipose tissue. This uncoupler keeps them warm. And another thing is 2,4-DNP or 2,4-dinitrophenol and aspirin. These 3 uncouplers, they cause they are the 3 main uncouplers, but the one that is in newborns is UCP or thermogenin in brown adipose tissue. All right. So does that make sense? Like you have to understand the difference between uncouplers and inhibitors. That's a common question that is asked. It's not asked directly, obviously, but you have to understand what this does and what this does to be able to know what the question is asking. What the question is asking for it. Yeah? So do you have any questions? Is that clear for uncouplers? Okay. And then the last point is okay. So obviously we talked about oxidative phosphorylation. We talked about obviously it has a lot of proteins, it has a lot of enzymes. Now, many of these enzymes, obviously, we know like in our body, we have the nuclear DNA and we have mitochondrial DNA. Now, 13 out of 90 proteins for oxidative phosphorylation are made by mitochondrial DNA and not the nuclear DNA. And why is that important? Because the mutation like mitochondrial DNA is more likely to have mutations than nuclear DNA. It's 10 times more likely. And for that to happen, like very important point is we know the nuclear DNA, it comes from obviously the mother and the father. Okay. However, mitochondrial DNA only comes from the mother. So it's maternal. So M mitochondrial and maternal. So M maternal and mitochondrial. The paternal one will be degraded. Okay. So that is why when most of the time, for example, here are some diseases. Okay. Some diseases are due to mutations in mitochondrial DNA, and mitochondrial DNA comes from the mother and not from both. Nuclear DNA comes from both. And because it comes like mitochondrial DNA only comes from the mother, and it also has a lot of mutations, like it's more prone to mutations. As a result, it causes what we call the mitochondrial myopathies. And mitochondrial myopathies, we have 3 main ones, which is MELAS. And MELAS, like I remember it came up in one of our exams last year. So you have to know MELAS is one of the mitochondrial myopathy because of mutation in mitochondrial DNA. Okay. Obviously red muscle fiber disease and Leber hereditary optic neuropathy. But the main one you have to know is MELAS. MELAS is very important. And as I said, it did come up in our exam. Okay. So yeah, that's the whole point of mitochondrial DNA, and because it codes obviously for the proteins in the oxidative phosphorylation, and it's more prone to mutation, and it comes from the mother and not from both mother and father. All right, so is that clear for everything? If you do have any questions, type it in chat, or you can unmute, whatever you want. Is that clear? Okay. All right. So I have like a couple of questions, about 3 questions. And I want you to volunteer, please. So all right, let's go. Okay. So the first question, who wants to read the first question? Does anyone want to read the first question?
I can read it.
Okay, go for it.
A laboratory analysis of isolated mitochondria demonstrates that oxygen consumption is normal when succinate is added, but extremely low when pyruvate and malate are used. The mitochondria are subsequently shown to have normal cytochromes, but a reduced iron content. Reduced pyruvate malate oxidation is due to a defect in which of the following respiratory components?
Okay, what do you think the answer is? Okay, so to understand it, remember when I said NADH enters the complex number 2, okay? But NADH, sorry, enters complex number 1. FADH2 enters complex number 2. So what it says here is that pyruvate and malate are being used. It's extremely low when pyruvate and malate is used. However, when succinate is used, which is complex number 2, which is also called succinate dehydrogenase, I want you to keep this in mind. Succinate dehydrogenase, which is a TCA cycle, which is basically succinate to fumarate, is also in the electron transfer chain. Okay, so here there's normal succinate. So complex number 2 is fine, right?
Yeah.
Okay, because obviously it's normal when succinate is added. So complex number 2 is fine. However, it is low when pyruvate and malate are used. So because it's low in pyruvate and malate, which is mainly pyruvate. We talked about pyruvate and NADH. So as a result, complex number 1 is not normal. So as a result, the oxidation is due to a defect in which of the following, which is complex number 1, right?
So succinate dehydrogenase.
Okay.
Oh,
What did you say?
I'll write the NADH one.
Okay, for complex number 1, NADH dehydrogenase. Complex number 2 is succinate dehydrogenase. Okay.
Oh, yeah.
Because it's asking for the defect. The defect is in succinate dehydrogenase. It's in NADH dehydrogenase. Because when you add succinate, it's normal. So it's not defected. It is normal. However, when you add pyruvate, it's not normal. That is why it's in complex number 1. Okay.
Okay, but to be clear, succinate is the one in the second complex.
Yes, because obviously we remember when we said complex number 2, what is it also called?
The FADH one.
Succinate number 2 is also called succinate dehydrogenase. Okay.
Yeah.
Which is okay. And succinate dehydrogenase, it has FADH2. Remember that? FADH2 enters the complex number 2, and when it enters complex number 2, obviously it causes like the entire electron goes to coenzyme Q, then 3, then cytochrome C, and then 4, right? So succinate, when you add succinate, oxygen is normal. However, when you add pyruvate and malate, complex number 1, like the oxygen consumption was not normal. So like the entire chain, it did not form, or it wasn't yeah, so technically you can say it was inhibited. So as a result, there's a defect in the first one, which is pyruvate and malate, which accepts pyruvate, the NADH from pyruvate and malate, which is succinate
NADH, the first complex.
Yes.
Okay.
Okay. Second question. Okay, who wants to read it? Anyone want to read it? Okay, fine, I'll read it. Okay, so you're studying the effects of a compound and respiratory activity of an isolated mitochondria. Your experiments demonstrate that oxygen consumption, okay, increases as expected when pyruvate and malate are used, as well as when succinate is added. So complex number 1 and complex number 2 are fine because oxygen consumption increases as expected. So it's normal. Okay. However, addition of a test compound, so this is the main point of the question, shows a significant rate, increase in significant rate of oxygen consumption. So oxygen consumption increases significantly, but it's not due to complex 1 or complex 2. Okay. However, so remember when we talked about inhibitors and we talked about the uncouplers. So if you look at the choices, remember what with the uncoupler was. Remember what was the difference between uncoupler and inhibitor? Inhibitor decreases oxygen consumption, decreases ATP synthesis, and decreases NADH oxidation. However, uncoupler, it decreases ATP synthesis, but it increases oxygen consumption. And that is why you're going to look immediately, you're going to look for the uncoupler. And that is why it is indeed D. Yes, it's dinitrophenol, which is one of the uncouplers. Okay, the rest, which is carbon monoxide, cyanide, and rotenone, are all
inhibitors.
They're all inhibitors, exactly. Okay, that makes sense. Okay, the third question. Okay, the third question. Does anyone want to read it, or should I read it again? Okay, I'll read it. So you're the physician examining a 6-year-old boy who has been experiencing progressive muscle weakness. You perform assays on muscle biopsy tissue from this patient and find that mitochondrial oxidation of succinate is normal, but pyruvate oxidation is severely impaired. Okay. Mitochondrial extracts demonstrate that the activity of pyruvate dehydrogenase complex is normal, as the activity of malate dehydrogenase. So these results are consistent with patient harboring a defect in which of the following mitochondrial components? Okay, so yeah, it is B. So it's exactly the same as this. It's exactly the same concept as number 1. But the point of this question, why I put it, is I want you to know the both names for every single complex, and I want you to understand again the concept as in pyruvate. Obviously, when like pyruvate oxidation is normal or a succinate oxidation is normal. Okay, so succinate oxidation is for like complex number 2. Pyruvate is for complex number 1. Okay, so indeed it's B. All right. So do you have any questions? I think we're done. Do you have any questions? If you do have, like, just type in chat. If you don't, then let me know too. All right, sounds good. Thank you. Thank you for attending. I hope you did benefit. Good luck.